Echolocation is the biological ability of animals to use sonar to locate objects in the environment. This ability is notably present in bats and dolphins. It should be obvious that this ability evolved convergently in both species. However, did they truly arise from selection pressures as evolution suggests, or did they evolve from mutations within the same genes? This research seeks to answer that question by analyzing and comparing the genomes of animals that independently evolved echolocation, mainly dolphins. Specifically, 2 echolocating and 2 non-echolocating bats were genome sequenced. In addition, the sequenced bats were aligned next to dogs, dolphins, rats, and humans. If echolocation truly did evolve from the same genes, we could study the same genes in other animals. Such studies could help repair deafness in humans if the same genes were present.
So some of the results were to detect genome-wide sequence convergence between echolocating lineages. The research group built an analytical pipeline based on maximum likelihood, which is the ML, phylogenetic reconstruction. Using this, they could analyze each amino acid and force echolocating taxa into erroneous monophyletic clades to represent different convergence hypotheses. The lab quantified the extent of sequence convergence at each locus by taking the mean of its SSLS, which means the site-wise log likelihood support. They found that a convergence of 824 loci for hypothesis 1, which was the bat-to-bat convergence hypothesis, and then the 392 loci for hypothesis 2, which was the bat-to-dolphin convergence. The team ranked the mean of SSLS for all 2,326 loci and both convergence hypotheses and inspected positions of hearing genes that exhibit convergence and adaptation in echolocating mammals. To name a few are Preston, TMC1, KCNQ4, PJBK, otoferlin, PCDH15, and CDH23. Besides examining the positions of hearing genes, the lab examined vision genes as well. They found 4 such loci in the top 5% of the genes in the bat-to-bat convergence hypothesis, and then 3 in the bat-to-dolphin convergence hypothesis. Comparing hearing genes to other genes, the team tested whether the hearing genes had greater statistical sequence convergence than other hearing genes. After comparing hearing and deafness genes, they found that hearing genes had significantly more negative average values than expected by chance for bat-dolphin convergence. This was repeated for 75 other genes listed in the vision blindness category. There are 5 solute carrier proteins related to Preston. It's a motor protein that drives the cochlear amplifier. They proposed that the loci with sites undergoing diversifying selection of convergence, adaptive convergence, would exhibit a negative correlation, whereas those loci with sites undergoing divergence driven by diversifying selection. But under the accepted species topology, adaptive divergence basically would exhibit a positive correlation. For the bat-to-bat convergence hypothesis, they classified 92 loci as putatively adaptive convergent and 111 loci divergent. With the same approach for the bat-dolphin convergence hypothesis, they classified 59 loci as adaptive convergent and 212 loci as adaptive divergent. This means that the larger the number of adaptive divergent loci for the bat-dolphin convergence hypothesis reflects the stronger average relative support for hypothesis 1. Using the fitted regression models for each locus, ultimately genes associated with hearing and vision, or those associated with deafness, showed significantly stronger convergence than the background loci.
Okay, so in conclusion, the study aimed to study the genomic relationship between the development of echolocation in bottlenose dolphins and 2 different bat lineages. By looking at the alignment of specific orthologous coding gene sequences across the entire genomes found in the studied lineages, researchers found extensive convergent genetic changes. The convergent genetic changes are found in genes that are related to vision and blindness mainly, as well as hearing and deafness. These genes may be associated with the shift in primary sensory modality, so basically a shift from vision to location, or echolocation. The study is significant because it provides strong support for genome-wide convergences, unlike many studies which report sequence convergence in only a few candidate genes. Work with these findings are currently underexplored and underexploited, so there's a lot of opportunities for future research that could be done regarding other homoplasies, or possibly at the molecular RNA level in an experimental fashion. Perhaps a study regarding the camera eye, which evolved convergently in cephalopods and mammals, could yield medical advances for humans.